Hi everybody, I hope you're keeping very dry because there's a lot of rain going on right now. This is Thursday, August 20th, and it's about 5:40. So the purpose of this assignment is really to have you guys read and practice data analysis. I know that you're doing presentations, so I kind of wanted to give you a preview of what that's going to be like, kind of get your feet wet a little bit. Some of you have already quite experienced with reading papers and feel comfortable with it, and maybe some other people aren't as much, and so this is just an opportunity to give you guys some practice. I'm going to give you some background information on this paper. This is a paper that I did more than 20 years ago, amazingly enough, during my my pre-doctoral thesis production. And so it's looking at shark antibodies, because I did work in shark, I told you guys that, and of a type of an antibody that we discovered early in development. So your assignment will be typed. You can work in groups of two, so I'm kind of showing you the document right here. You can work in groups of two if you'd like, or you can work by yourself, whatever you'd like. Please make sure that everybody's name is on the report if you are going to work in a group of two, and also only one member of the team should submit the report. That's right here. And please remember that all of these reports are using the Turnitin anti-plagiarism tool, so that if you Turnitin and you submit your report and you get a high similarity score and it's colored in red, that's not good. You're going to have to revise it. That means that you've either inadvertently or purposely copied from somebody else. So just fix it, revise it, resubmit. I told you there's an unlimited number of submissions that you could do. I've given you the rubric of how I'm going to score things, so it's quite clear, and then I'm going to go ahead and start now with the background information. So in the first figure that I'm going to go over kind of as a practice, it's really looking at proteins which are in the blood, which were separated out from, because we separated out the cells of because sharks are just like mammals, all right? So they've got red blood cells, they have white blood cells, they use oxygen to breathe, and so they have, if you separate out the cellular component from the liquid component, that's what's left behind without cells is called plasma. So in the plasma, there's all kinds of proteins in addition to that help with osmotic pressure, such as albumin. They also have like clotting factors. Then you have other proteins that are in the blood, in the plasma, and that would be the antibodies. Antibodies are there because they help protect the organism from infection and from disease. And so that's part of the adaptive immune response. So we're looking at proteins, antibodies specifically, that are in the plasma of shark. And so that's why I kind of gave you, what is plasma. Now sharks kind of have a difference in antibodies. So everybody's got IgM. So IgM is kind of like the first type of antibody which is made, relatively large molecule. And in humans and most mammals, so over here, if you look at this, it's a pentamer or a hexamer. In sharks, it's going to be a pentamer or a hexamer. That means there's either 5 IgM molecules which are tied together using a J chain protein, or 6. And that's a relatively large structure, because it's pretty much, it's got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 binding sites for something that those antibodies would recognize. So kind of very good at recognizing what it was built for. And but it's so large, it really cannot get outside of the vascular system and go into the tissues. So there's another kind of form of IgM which is made in sharks, and that's called the 7S. So these are kind of the sedimentation rate. So a very large molecule would sediment fast, and a very small molecule would take longer to sediment using ultracentrifugation. So it's a smaller molecule, 7S, and so that's the one that seems to get into the tissues and go and move around. All right, so I've given you some references if you want to read more about IgM and in shark. you are welcome to read that. And also, it's relevant because of our findings. We're kind of like piggybacking on top of what they found. Our findings were, of course, much more recent, and some of these findings were going all the way back to the early 70s, late 60s, so pretty long ago. All right, so the first figure that we're going to practice is taken from the paper that was published in 2001. Oh my God, that was like so long ago. And it's got a very long title, and if you read that publication information that I gave you in the Module 1, it's a shark antibody heavy chain encoded by a non-somatically rearranged VDJ, is preferentially expressed in early development and is convergent with mammalian IgG. So there is no IgG in shark, but this might be a nice precursor to IgG. So that's a specific type of antibody which is made. That can work really well with detection and the removal of something that is foreign. And most antibodies, all antibodies, at least in mammals, are rearranged at the somatic cell level. So not in the germline cells, in the somatic cell level. So, and that's why you can have such a large variety of antibodies in your blood. And right now, that antibodies are really, hot in the news, because we're looking for antibodies to be formed against COVID-19, against the novel coronavirus too. All right, so I've given you a partial information about the animals. These were newborn animals, and we're also looking at some adults, and so some information about that, and then also some information about monoclonal antibodies. So that's a specific type, a pure specific type of an antibody which recognizes one thing. And then immunoprecipitation is how you can use antibodies to pull materials out of, let's say, tissues, or recognize a material on a cell or in plasma in this case. Metabolic labeling, so this is using S35. S35, that's sulfur, is going to be present in amino acids which are going to be methionine and cysteine. So if you label cells when they're producing the antibodies, you will actually radio-label the antibody, the amino acids of methionine and cysteine, and that can be then detected, can be used to detect information using development of films. Okay, so here we have the figure that I wanted to just go over with you as a practice. And so we've got, these are PAGE gels. This is polyacrylamide gel electrophoresis, and that's what PAGE stands for. These are used to analyze proteins. In this case, we're looking at plasma, and the sizes of the proteins will migrate according to largest to smallest. So if they're the top of the gel where these numbers are, that's going to be a relatively large molecule, and as they migrate down the gel, they will migrate faster if they're smaller. So it's all based on size, so it's separation by size in this PAGE gel. We have it under non-reducing conditions. That means if things are bound together, they'll stay together, and then you can add some other materials to kind of reduce the proteins, and that will separate, that breaks the bonds that we're kind of having them form together, and you'll see the separation. And the reason why we do this is because antibodies are always composed of a heavy chain and a light chain. So when you keep the heavy chain and the light chain together, you're under non-reducing conditions, and when you separate out the heavy chain and the light chain, that's what we've done that using a reducing material. So here's the large, pentamer of IgM, and let's see, 1, 2, and 3. So 1 is going to be, let's make sure. So that's an antibody which is a negative control, is in lane number 1. And if it's a negative control, you don't expect it to interact with any particular proteins which are going to be in shark plasma. The second lane is a type of antibody, monoclonal antibody, CB16, and here it's explained what it reacts with. It's IgM specific, so it's specific for IgM, but it does not cross-react with a particular type of IgM that we found in the young animals, and that's called IgM1GJ, and that means, I'm sorry, it's so, we used to call it baby IG, but then we had to get a little bit more, you know, professional. But it means that it's the first IgM gene which is expressed during development in primary lymphoid tissues, which are going to be epigonal organs in the shark, and GJ means germline joint. So that means that this is kind of a pre-rearranged antibody, which is rather unique. It has come up before, but it's the first time we've actually detected it specifically, and we actually found the genes as well. So here we've got, under non-reducing conditions, you've got this multimer of IgM, and this is also in these animals here. This is all neonatal plasma. So this is all plasma from newborn babies. So you've got some IgM that's in the large multimer, and then you've got a relatively large amount. So this antibody, lane 2, doesn't cross-react with the IgM1GJ, but the lane number 3 is GA15 monoclonal antibody, and it is IgM specific, and it does cross-react. So recognize this IgM, there's your IgM up there, and also a smaller type of an IgM called IgM1GJ down here. And so that's, we're detecting that. And then if you take that material and you reduce it, you can see that there is a heavy chain up here. That's what the H stands for. The heavy chain for the 1GJ gene is smaller. It's shorter and smaller by 1 domain. So it's physically smaller in size, and that's why that band is lower in the gel rather than higher. And then it is associated with a light chain, and that's why you see this dark band down here. So it looks like a regular IgM, but smaller in size. And that is kind of the analysis. So this is just looking at plasma in newborns. It is not comparing that information at all to the adult. That comes up in the next step. So here we've got newborn animals, and we're comparing that to adult animals down here in B and C. And that is for you now to look at. So I'll just let you know that the epigonal organ is kind of like the bone marrow equivalent in sharks. So they don't have bone marrow because they're cartilaginous, so they do not have bone marrow, but they do have a substitute organ that's called the epigonal organ, which we think is kind of the bone marrow equivalent. And then it has a spleen just like you and I, and this is also where immune responses have been shown to occur. So here you're going to look at this, and you can see there's different antibodies that are being used, and then there's information here. And so what I wanted you to do was to kind of look at this, answer these questions, and then you write your report, and then you submit that. So hopefully that explains everything to you guys, and keep dry. And I'll talk to you later. If you have any questions, just shoot me an email or give me a call. Just make sure that you let me know who you are when you call me. All right, bye.